Synthetic biology is an exciting field that offers innovative approaches for engineering new biological systems or redesigning existing ones for useful purposes. As a disruptive technology, synthetic biology is capable of solving complex global healthcare challenges, such as the organ shortage crisis. The advent of organ transplantation in the mid-20th century revolutionized medicine and gave those suffering from end-stage organ failure a second chance at life. As surgical techniques improved, the number of diseases treatable by organ transplantation has steadily increased over the last 30 years, resulting in a huge demand for donor kidneys, hearts, livers, and lungs. The majority of transplanted organs are harvested from recently deceased individuals who have consented to the procedure prior to their death. Unfortunately, the supply of viable organs from deceased donors is insufficient to meet the increasing demand. By some estimates, as little as 5% of the organs needed become available. As a result of this shortage, an average of 18 patients on the National Transplant Waiting List die each and every day. Given our aging society, this trend is set to continue. Consequently, alternative methods for obtaining a steady supply of transplantable organs are needed. One of the more promising solutions is xenotransplantation. Xenotransplantation refers to the transplantation of animal cells, tissues, or organs to replace an injured part of a human recipient. Xenotransplantation is not a new concept and was first attempted, albeit unsuccessfully, using rabbits in 1905. Undeterred by failure, surgeons continued to attempt to transplant organs from various non-human primates throughout the latter half of the 20th century, reasoning our close phylogeny with apes would improve the likelihood of success. However, it was soon realized that primates were not suitable animals for xenotransplantation due to organ size disparities, risk of cross-species transmission of infection, and difficulties in breeding. Instead, the most compatible animal was found to be pigs. In addition to being prolific breeders and quick growers, pigs also have surprisingly similar anatomy and physiology to humans. Moreover, the large variety of pig breeds allows the size of the organs harvested to be matched to the recipient. A form of pig-to-human xenotransplantation already exists: porcine bioprosthetic heart valve replacement. Since the 1960s, pig valves fixed with glutaraldehyde have been used to replace damaged human heart valves with reasonable success. However, the ultimate goal is to be able to transplant entire organs. Despite our similarities, genetic discrepancies between humans and pigs inevitably results in immunological rejection, whereby the recipient's immune system recognizes the transplanted organ as a foreign invader and destroys it from the inside out. This process is swift, and in the case of hyperacute rejection, can occur within minutes of transplantation. Hyperacute rejection is a violent reaction that is mediated by antibodies that naturally pre-exist in the recipient's body. These preformed antibodies bind xenoantigenic proteins that protrude from the surface of porcine endothelial cells and trigger the activation of complement proteins. Activated complement will go on to lyse these foreign cells, thereby disrupting the vascular integrity of the donor organ, leading to its failure. Whilst immunosuppressive therapy can dampen the host's immune response to a degree, it is a stopgap solution that doesn't address the underlying cause of xenograft rejection. An alternative approach to reducing antibody-mediated mechanisms of rejection, with demonstrated success, is to humanize the organs transplanted from pigs using the tools of synthetic biology. For example, the main xenoantigen responsible for hyperacute rejection is galactose alpha-1,3 galactose, or alpha-gal for short. In human blood, approximately 1% of all circulating antibodies are directed against alpha-gal. Therefore, in order to obtain transplantable organs from pigs, the alpha-gal antigen must be removed from all cell surfaces. Using homologous recombination, Day et al. Were able to disrupt the glucosyltransferase gene responsible for adding alpha-gal to surface proteins of pig fibroblasts, thereby preventing human serum from eliciting an immune response. The researchers then used those fibroblasts to produce alpha-gal glucosyltransferase knockout piglets by somatic cell nuclear transfer, whereby the nuclei from the donor cells were implanted into empty egg cells. Whilst knocking out alpha-gal alone is not enough to convince the human body to accept a whole organ from a pig, more extensive genetic engineering could make xenotransplantation a reality very soon. For instance, using CRISPR-Cas9, Yu et al. Were not only able to knock out 3 different xenoantigens in pigs, but also introduce 9 human transgenes designed to inhibit complement activation and enhance immunological compatibility with humans. Although the technology is still in its infancy, organs made to order using genetically modified pigs may one day save your bacon.